Protein verification. It is important to verify that the target protein is not lost during each stage of purification. So basically, first thing you do is SDS page. You can use a detergent. How it made its detergent SDS is added to the protein mixture, and mixture is heated. These sulfate charge are evenly distributed along the denatured protein, making separation dependent on size and not on the charge. After treatment with SDS protein sample, is loaded into BAGE, which is a polyacrylamide gel electrophoresis, where it forms a single band at a specific location on the gel depending on its molecular size and mass. So how you visualize the proteins after running on SDS page? The straightforward is to use a Comassi stain. It's a dye that combines with protein, so a color band results. Usually it's blue in color. You can compare the stain sample with known size and marker to estimate the protein size. During purification process, the colored band representing the protein should become increasingly intense, showing that its protein is not lost during the procedure. So in other words, during the purification procedure, if it's going okay, so that means during the purification you have the band will show increasing color. Okay, so we can see that in the figure. This is the electrophoresis system used for electrophoresis, and you can see from the top figure you can load a protein into the well. So this is how it goes when you run the sample. You can see you have 5 different stages of purification, and in lane 5 you have the desired protein only have been separated from the other component of the protein that came with the cell lysis of the organism. You can see this actually figure showing the protein have been isolated, and now it's clear in the sample. Another way of verification is the Western blotting, and it is a specific method for protein separation used after doing SDS page. So normally proteins after SDS page are transferred to nitrocellulose membrane using electrical current. All sites on the membrane are then blocked so that the antibody will not bind to them non-specifically. You put a primary antibody that is specific to the protein of interest and you incubate with the membrane. If there are any antibodies present that are directed against one or more of the blotted antigen proteins on the membrane, this antibody will bind to the protein and the other antibody will be washed away at the end of the incubation. That means if you got your antibody, if you got your protein of interest and you incubate with the primary antibody, it will bind. If there is no protein, the antibody will not bind. To detect the antibody that have been bound to the protein, a secondary antibody is used coupled with a reporter, which are usually alkaline phosphatases are added to the membrane. Excess secondary antibody as well is washed off the membrane. Then the substrate is added to this antibody, which is linked with alkaline phosphatase reporter group. Upon reaction, a visual band will appear indicating the presence of antibody. And you can see here, this is a readout of the Western blot. You can see that you have higher molecular weight than expected, often due to glycosylation. So glycosylation is the addition of carbohydrate group on the protein. Once the same protein have an additional carbohydrate group, it will migrate, it will be retarded because the migration is according to size. The third lane, it shows low molecular weight protein than expected, usually due to cleavage or degradation. So actually all of them, 1, 2, 3 are the same protein, but the normal one is in the lane 1. No, number 2 migrated slowly because it's glycosylated, so it's heavier in size, while the third is a little bit degraded, so it's smaller in size. The other method for protein verification is ELISA. It's a very sensitive, practical assay useful in detecting protein even at low concentration. You have a direct ELISA where the antigen or your protein of interest is coated on a glass or a membrane, and you have the primary antibody tagged with a reporter group that gives the signal once the substrate is added. You have indirect ELISA. Actually, in indirect, you need a secondary antibody conjugate with a reporter that gives the signal with a substrate. You have a sandwich ELISA. You need in this case, in sandwich ELISA, the scaffold is having antibody which is specific to your protein of interest, and then you use the primary antibody and then the secondary antibody conjugated to a substrate that gives signal, conjugated to a reporter that gives signal with the substrate. Then you have the competitive ELISA. That means you have the antigen or the protein of interest coated on the substrate, and then you incubate with two different antibodies. One is specific to your antibody and the other is not, and it only will give a signal when the right antibody binds to your protein of interest. So in all these four cases, giving the resultant signal is indicative of the presence of your protein of interest. So after you verify protein, you need to preserve protein. Once the protein of interest is isolated, collected, purified, it must be saved to preserve its activity. It can be lyophilized. Freeze drying is the best choice because it maintains the protein structure. The post-purification analysis methods include protein sequencing, of course, to know the sequence, mass spec, of course, to know the unique signature and what is this protein. The methods include as well X-ray crystallography. This used to determine the tertiary and quaternary structure of protein, and this requires a pure crystal of protein that have been dehydrated from solutions. When exposed to X-ray, a pure protein creates a specific shadow pattern based on its configuration. So using a computer analysis of the pattern allows the description of the structure of proteins. The fourth subtopics is proteomics. Actually, proteomics is a scientific discipline dedicated to understanding the complex relationship of disease and proteins expression. Proteomics are compared between healthy and diseased states. The variation of the protein expression are correlated to the onset of progression of disease. The goal is to discover protein markers to be used as a diagnostic method and development of a target therapy or target drug. The techniques used for proteomics, it's a two-dimensional gel electrophoresis separates a protein followed by a mass spec to verify the protein identity. They can use amino acid sequence to characterize the protein. Protein microarrays can be used as well to set a protein immobilized on a surface coated with a reagent that will indicate binding by color change. Functional protein microarray have been recently applied to discovery of protein interaction, protein carbohydrate, protein lipid, protein DNA, protein drug, and protein-protein interaction. Thank you.